Well, hello class. Welcome to everyone. Today we are going to discuss fertilization. Okay, so in the previous lecture we have already covered the menstrual cycle and the gametogenesis. Okay, both spermatogenesis and the oogenesis. Today in this video lecture we are going to cover the process of fertilization. Okay, so let's begin. What is fertilization, right? So we have been reading this since lower classes, that the process of fusion of gametes to form zygote is called fertilization, right? Now here what we are going to emphasize is the detailed procedure, how the two gametes reaches to each other, and when they meet what happens with them. Okay, how they make sure that only the compatible gametes will fuse, and how their nucleus fuses to form the zygote. This is all we are going to see in detail. Great. Well, so let us begin with the sexual act, right? So the sexual act between an adult male and a female is called coitus. Okay, so that is called coitus. Right? So it is a sexual act or reproductive act between an adult human male and a female. Right? Now during coitus, what happens is the male deposits the semen into the female's body with the help of external genitalia. Okay, to be precise, with the help of penis, right? So this is the exact function of the external genitalia, that they help or they assist in the transfer of gametes, okay, or collection of gametes. So with the help of external genitalia, to be precise penis, the male deposits the male gametes, not only male gametes, it deposits semen that contains male gametes into the female genitalia. Okay, now where does it do so? So during coitus, let us see. So what is happening here is, during coitus, the male is depositing semen into the vagina of female, right? Great. Now, so can you see, this was the part of vagina, the deeper part of vagina. So here the male is depositing the semen, so inside the vagina, and that's why this process is called insemination. Okay, so what is insemination? The process of depositing the semen inside the female genital tract or into the vagina is called insemination. Great. Well, very good. Now we have already seen, we have studied the composition of semen, and we know that in one ejaculation, okay, the semen contains on an average about 300 million sperms, okay, or spermatozoa, right? So what actually is happening, the male is depositing around 300 millions of sperms into the female body, okay, and this whole process is called insemination. When it is happening, it is happening during coitus, right? Great. Now story begins from this place. Okay, so this was the foreground. So from here the story begins. Let's see. Now, once this 300 million sperm, okay, we are taking this average number. This number may be more than that, this, or can be less than this. Okay, it again varies from person to person. We are not going to take into account that. We are sticking to the average number. 300 million sperms are being deposited into the female body, into the vagina precisely. Now what happens? Is it like all 300 will start the race? No, not at all. Now, most of the semen, okay, sperm is in the semen, right? Now, most of the semen will flow out. They will have a backflow and they will flow out. So, most of the semen either flows out or most of the sperm in the semen dies because of acidic nature or acidic medium. Okay, so inside vagina the condition is acidic, and because of this acidic condition a lot of sperms are going to die. Okay, most of them are going to flow out. So majority of the volume of semen that is deposited during coitus flows back, okay, so it flows out of the female body. Only some amount goes up, okay, remaining flows out, and whatever remains there, now out of that most of the sperm are going to die because of the acidic conditions, right? So obviously not 300 are going up, not at all. Only a few millions are going to go up. Okay, remaining are going to be either flown out or die because of acidic medium. Great. Now what happens? So a few millions will be able to cross this cervix. Remember the cervix? Now the cervix usually remains tightly closed. Okay, and inside the gap in the cervical canal a thick mucus is filled, that is called cervical plug. But during the days of ovulation, okay, so about 3 days, okay, of ovulation, or 3 days period around ovulation period, the cervix opens slightly and the cervical mucus present in that also gets diluted. Okay, as a result, now the sperms can swim through this viscous fluid, okay, and get past the cervix. Now once this period is over, it is practically very difficult for the spermatozoa to cross this cervix. Okay, great. So we are considering that it is the time of ovulation, okay, and the cervix is open. The mucus inside this cervical canal is thin, so around few millions of sperms will be able to cross this cervix and ultimately reach into the uterus. Okay, now inside uterus what is happening is the walls, okay, the walls have a lot of macrophages. Okay, so now a lot of sperms will be eaten up by the macrophages. Okay, so here there are macrophage which are going to do what? So they are going to phagocytize, or they are going to do phagocytosis of a lot of sperms. So now a lot of sperms are again going to die because the WBC present in the uterine wall are going to do phagocytosis. Well, not only this, now a lot of sperms are also going to die. Okay, die because they will run out of food or out of fatigue, so they are going to die. Great. Finally, what happens? Only around few hundreds to few thousands. Can you see? Few hundreds to few thousand of sperms are able to reach into the fallopian tube. Now imagine, we started with 300 million, right? And only few thousands at max are reaching to the fallopian tube. Can you see? So more than 1,000-fold are lost. Okay, now great. What happens once these few thousands reaches the fallopian tube? Now they start to swim. Okay, Now you will say, sir, in the uterus you said a lot of sperms die because they have lost their vigor, they cannot further swim, or they are being phagocytized. Now once a lot of sperms have lost their vigor, how all of a sudden they are going to get a lot of energy to swim into the fallopian tube? Well, what happens here is a unique process called capacitation. Okay, what is happening is capacitation. Do remember this word, capacitation. Now what is this? When these sperm are in the uterine wall and finally reaches the fallopian tube, now there are a lot of chemicals, okay, chemical signals present into this fallopian tube and uterine part of near the fallopian tube that alters the acrosomal membrane permeability. As a result, the sperm becomes hyperactive. Okay, hyperactive both in terms of its swimming, the speed of swimming, okay, as well as their vigorness. Okay, so these sperms now become more vigor and they swim harder and faster. Great. Now finally these sperms, few thousands of sperms which are swimming into the fallopian tube. Now the fallopian tube is lined by ciliated epithelium. Okay, and most of these sperms are going to get stuck into this cilia. Okay, so if you look into the fallopian tube, it will appear as if there is a jungle of grass, okay, tall grass into the fallopian tube. What are these grass? Actually these are cilia. So most of these capacitated sperms are going to get stuck into the cilia and then they are going to die out there. Okay, only a few dozens, okay, only a few dozens. Okay, अब आप कहाँ बात कर रहे हो? Around 20, 24, 25, okay, or at max 36 or so. Only a few dozens of sperms are going to win against all these odds and finally reach to the ovum. Okay, so remember we started with 300 million sperms and only a few dozens of them were able to make up to the ovum. Okay, and therefore we say that the journey of sperm is quite challenging. Okay, a lot challenging, right? And now you know why so many of sperm is required, because most of them are going to die on their way. Only a few are going to reach up to the what? Ovum. If the number of sperm will be less, then the likelihood of any sperm reaching up to the ovum into the fallopian tube, to be precisely the ampulla of fallopian tube, will be negligible or no. And that's why a lot of couples which have low sperm count, they have difficulty or they fail to conceive naturally. Okay, great. So this was the one-sided story. This was the story of male gamete. Let's look at the female gamete. Okay, now meanwhile what happens, look at here, I'm going to zoom a little bit. Okay, so I'm zooming here. So if you see, this is the ovary, right? And at 14th day ovulation happens. So this ovum comes out of the Graafian follicle and the ovary and finally released into the body cavity. Okay, now once released, this ovum is catched by the help of this fimbriae at the end of fallopian tube or oviduct, and they will make sure that this ovum enters into this fallopian tube, right? And finally this ovum enters into the fallopian tube, the infundibulum part, which is trumpet shaped, and with the help of cilia. Okay, so I already told you that the wall of oviduct is lined by ciliated epithelium. So these cilia keep on beating, and they beat in such a way that they pull in the water. Okay, so they beat in such a way that water will go in. Okay, so water will go in. As a result, even the ovum along with water will move inside. Okay, great. So this is how the cilia beats, and this is the function of cilia here, to assist the movement of ovum. Finally, what happens? The ovum moves through the infundibulum and finally reaches into this widest part called ampulla, right? I hope you all remember the parts of oviduct. Now, as the ovum reaches ampulla, now it slowly starts to cross the ampulla. Okay, now at the junction of this ampulla and isthmus, precisely this point, this is the junction of ampulla and isthmus, or you can say maybe this point. Okay, so this is the junction of ampulla and isthmus, and this is the point where the sperm and the ovum meet with each other. Okay, and therefore this is the site where the fertilization takes place. Okay, so do remember, it is important. This is important. Okay, so site of fertilization is the junction of ampulla and isthmus. We also call it as isthmus ampullary junction. Okay, isthmus ampullary junction. याद रहेगा? Great. So it is a very common question. The site of fertilization in oviduct is the junction of ampulla and isthmus, or isthmus ampullary junction, right? Great. So this you know now. Now what we are more interested in studying is how this fertilization happens, right? So let's look at in detail. Well, so here I have made a diagram for you. Now, if you look at this diagram, you know that this structure, okay, is called ovum, right? You remember the structure of ovum? The ovum had secondary oocyte and first polar body, then there was perivitelline space, right? And finally above to that was the zona pellucida, and on the surface of zona pellucida there were the cells of corona radiata. Correct? So this was the ovum, the structure of ovum. Now see, so consider that this is the structure of ovum, and these cells are the cells of corona radiata. Okay, great. So these are corona radiata, and then this blue layer is zona pellucida. Okay, so this blue layer is zona pellucida, and then the gap in between this space is called what? Perivitelline space. Perivitelline space, and the black one is the secondary oocyte membrane, right? That is secondary oocyte membrane. This was the structure. Great, you remember this. Now let's see what happens. As the fastest sperm reaches to the ovum, remember the fastest, okay, or the first sperm to reach the ovum, right? What happens? It passes through the layer of corona radiata, okay, and in order to get through this layer of corona radiata, it releases its acrosomal enzymes, right? Now, when it releases its acrosomal enzymes, these acrosomal enzymes are hydrolytic in nature, okay? They are hydrolytic in nature, and as a result, they dissolve the corona radiata layer of that area, okay? What happens? Finally, at that point, there is some actin processes that are made, okay? So look at this now. Can you see? Can you see here? Great. So there are some actin processes, and these are the red ones are the enzymes of acrosome, which are hydrolytic in nature. Now, they will degrade this layer of corona radiata. As a result, the sperm head will finally be able to reach up to the zona pellucida, the layer of zona pellucida. Now, this whole process, the whole process of dissolving the corona radiata layer with the help of acrosomal enzymes is called acrosomal reaction. This is called acrosomal reaction. Now, after this acrosomal reaction, what is happening? The sperm is able to reach up to the zona pellucida. Now, on the membrane of zona pellucida, see here carefully, on the membrane of zona pellucida, there are these receptors, okay? And what are these receptors? These are called sperm receptors. These are called sperm receptors. Why we call them sperm receptors? Because these are going to bind with the sperm. Okay, so these sperm receptors are present on the membrane of zona pellucida. Okay, and they are crossing all the way through this vitelline space and going into the oocyte. Can you see? Great. Now what happens? See here, I'm zooming it further. So just a minute. So look at this point. One sperm receptor is going to bind with another receptor present on this sperm. Great. So finally, these two receptors are going to interact with each other. These two receptors are going to interact with each other, and this interaction is the basis of inbreeding in a species. Okay, so this interaction is similar to the pollen pistil interaction. Here the sperm receptors is going to recognize the receptor present on this sperm, and if this interaction is positive, then only this sperm will be allowed to enter into the zona pellucida layer. If not, this sperm will not be able to penetrate through this zona pellucida, and fertilization will not occur, and hence reproduction will not happen. Okay, are you getting me? So suppose the egg of one species is subjected to sperm of another species. Okay, and these two are totally unrelated, different species. Okay, which are totally unrelated, very different from each other. So even if the sperm of one species is able to dissolve this corona radiata layer of the ovum of another distinct species, the sperm receptor reaction will not be positive, thus interaction will not be positive, and as a result, the sperm of totally different species will not be able to cross into the zona pellucida, and fertilization will not happen. Okay, so this is the mechanism. Okay, molecular mechanism for inbreeding within a species in the natural conditions. Okay, anyway, that is another part. Let's see, here we are studying about the reproduction in human, and we know that, or we are going to discuss only about the uh, interaction between sperm and the ovum of the human beings. So this is same species, there is no chances of negative interaction. So what will happen, this sperm receptor will bind with the receptor on the sperm, and finally as a positive interaction between these two receptors, this sperm will be allowed to penetrate through the layer of zona pellucida. Okay, now let's see. Well, what you are seeing here in this diagram is a portion of the ovum. Okay, I'm not showing the complete ovum, rather I'm going to show you a portion of the ovum. Okay, and let's see how and what are happening next to the entering of the fastest sperm into the zona pellucida. Okay, so see, this was the fastest sperm. Okay, and due to positive interaction at the sperm receptor, this sperm was allowed to enter through the layer of zona pellucida. Okay, so this blue line is zona pellucida. So once it entered through this zona pellucida, now only this head part has entered, the tail and remaining part remain outside. Okay, now this membrane of the sperm is in direct contact with the cell membrane of the secondary oocyte. Correct? So the black one is the cell membrane or plasma membrane of the secondary oocyte. Now what happens? The membrane of the sperm and the secondary oocyte fuses with each other. Okay, so they are going to fuse with each other, and because of this fusion, now the head part, remember this part, the head part of this sperm, only this particular sperm, okay, not any other, will be taken inside. So it will fuse, it is like a membrane vesicle fusion. Okay, so the membrane of the sperm and the secondary oocyte fuses. As a result, the entire content of the head part of the sperm enters into the cytoplasm of the secondary oocyte. Can you see here? Now it has entered. This blue one is the compact genetic material of the sperm, and this is the centriole, and here there is a small amount of cytoplasm of the sperm. So sperm has brought into the secondary oocyte only these three things. What? Its compact DNA, genetic material, centrosomes, that is centrioles. Okay, so centrosome have centrioles, so it contains centriole, and some amount of cytoplasm. That's it. Nothing else is brought in. The tail and middle piece remain outside the secondary oocyte, and it is degenerated. Well, now once the first sperm has entered, once this has entered, there is subsequent changes into the membrane of the secondary oocyte. What happens? As this sperm enters, there is an influx of calcium 2 plus from the endoplasmic reticulum. Okay, so ER releases a lot of calcium. Okay, now in presence of calcium, what happens is these cortical granules. Okay, so inside this, can you see here? So yeah, if you see this ovum, on the inner side of this ovum there are a lot of cortical granules. Okay, so there is a layer of cortical granules which I have shown here. Well, so because of this calcium, now these cortical granules in presence of calcium go and starts to fuse. See here, they go and starts to fuse with the plasma membrane of the eggs, secondary oocyte, and they will release their content, okay, release their content into this perivitelline space. Okay, so all this content is released into this perivitelline space. Now in this content there is a particular compound, there is a particular molecule called zip. Now what is zip? Now zip is zonal inhibitory protein. Okay, zip, the full form of zip is zonal inhibitory protein, and this protein is responsible for the cleaving of sperm receptors. Okay, so what is happening once the first sperm cell has entered into the secondary oocyte, cortical reaction is happening. Okay, this is called cortical reaction. I'm writing it here. Cortical reaction. Okay, and during cortical reaction what happens? Upon the entry of the first sperm inside the secondary oocyte, there is a release of calcium from the endoplasmic reticulum. As a result, the cortical granules of the secondary oocyte go and fuses with the membrane of the secondary oocyte, releasing their content into the perivitelline space. Okay, now in this content there is a particular thing called zonal inhibitory protein, or ZIP. Okay, now this ZIP goes and destroys all the sperm receptors present on the secondary oocyte. Now once all these receptors are destroyed, now no any other sperm can enter into the secondary oocyte. Great. However, what if another sperm has already entered through the zona pellucida, okay, entered already into the zona pellucida, okay, or already it is entering into the zona pellucida. Suppose this is another sperm. So what happens? Now this perivitelline space, into the perivitelline space, a lot of carbohydrate is secreted, and this carbohydrate, okay, this absorbs a lot of water and swells up. So this area swells up. As a result, now the distance between the secondary oocyte membrane and the zona pellucida increases. Okay, this increases. As a result, now no any other sperm, even if it has crossed or is in the midway of crossing the zona pellucida, will be able to reach the secondary oocyte membrane. Right? Now, this swelled up perivitelline space and the zona pellucida now is called fertilization envelope or fertilization membrane. Okay, now in the fertilization membrane, the zona pellucida part becomes very hard. Okay, so now the zona pellucida become very hard. At the same time, all the sperm receptors has been destroyed, and the perivitelline space is filled with a matrix which has swelled up. Now all this together ensures that no any other sperm will enter into the secondary oocyte after the entry of first sperm. Okay, now why it is of so much importance that only one sperm enters? It is important because if more than one sperm will enter into the secondary oocyte, then the chances of making polyploid embryo or zygote is possible. Okay, so in order to maintain the chromosome number, in order to maintain that only diploid zygote is formed, it is mandatory to have a single sperm entering into the secondary oocyte, and it is ensured by the acrosomal reaction, by the cortical reaction. Okay, I hope आप लोगों को समझ में आ रहा है. Great. So this is what happens. This is called cortical reaction. Okay, now once this cortical reaction has happened, the membrane of the secondary oocyte or this ovum has hardened, and no any other sperm can enter into this secondary oocyte. Well, that is okay. Good. Now what happens? Okay, what happens with the genetic material of the first sperm that has entered into this secondary oocyte? How this fertilization will occur? Let us see. Okay, coming back to this image, now let's see. Considering that this was the genetic material that has entered from the first sperm into this secondary oocyte, correct? Now this genetic material of the first sperm is very compact. It is very compact. So what happens next here is this compact genetic material opens up. This starts to open up, and we call it swelling. So the next step is swelling of the genetic material of the sperm. So it swells, and finally with the help of centriole, this chromosomes or this DNA from the male gamete will condense to form chromosomes, and it will get arranged. Okay, and finally one nuclear membrane is formed around this male genetic material, right? So because of this whole process, what is formed is called male pronuclei. Okay, so the condensed genetic material from the sperm swells up and finally arranges itself in the form of chromosome, and a nuclear membrane appears around all these chromosomes of the male gamete, forming male pronuclei. Okay, so this male pronuclei is formed. Now what happens simultaneously into the secondary oocyte? Let us see. Remember, we have already studied that secondary oocyte was arrested in its prophase 2, right? It has almost completed its prophase 2 and has to initiate metaphase 2. But the secondary oocyte was unable to do so because it was lacking centriole. It was lacking centriole. But the sperm that has recently entered into this secondary oocyte has brought with it extra pair of centriole, right? And this extra centriole will go to the secondary oocyte and helps it to complete its meiosis 2, right? Now as a result of completion of meiosis 2, what will happen? 2 cells will be formed. Now let me make here. So this is one cell and this is another cell. This is meiosis 2 completed. So meiosis 2 will be completed with the help of centriole coming from the sperm. So male gamete centriole will help the secondary oocyte to complete its meiosis 2. Yaad rakhiyega. Okay. Secondly, this meiosis 2 is completed after fertilization when the sperm has entered into the secondary oocyte. Important hai ye. Please याद रखिएगा, ठीक है? Meiosis 2 of the female gametogenesis finishes or occurs after fertilization. Post fertilization होता है ये, ठीक है? Now because meiosis 2 is completed, meiosis 2 again is unequal division here. As a result, 2 cells will be formed. Okay, one larger cell which is called ootid or egg cell, and another smaller cell here which is called second polar body. Now this cell is called second polar body. First polar body was formed earlier. This is the second polar body. Okay, now if you look at the nucleus of this newly formed egg cell or ooted, now this nucleus is called female pronuclei. This is called female pronuclei. Finally, what happens? The male pronuclei advances towards the female pronuclei and fusion occurs. Okay, fusion occurs, and this fusion of male and female pronuclei is called syngamy. Is called syngamy. कहाँ हो रहा है? Into the ooted cell or egg cell. Remember? So after this syngamy, what will be formed? Will be a diploid nucleus, and this nucleus, okay, has homologous pair of chromosomes. That's why it is diploid, and it has one chromosome from father and another pair from the mother, and similarly for rest of the chromosomes. So if you look at, if humans have 46 chromosomes, that is 23 pairs. Every gamete has just 23, 1, 2, 3, 4, 23. So the male pronuclei will be having 1 to 23 chromosomes. Similarly, female pronuclei will also be having 1 to 23 chromosomes. And when this syngamy will occur, one from the male gamete and one from the female gamete will pair. Similarly, 2 from father and 2 from the mother, that is male and female gamete, will pair. So all these 23 chromosomes will pair up, and total 46 chromosome will be found in the zygote. This whole, now this ooted cell after fertilization will be called zygote, right? Now out of all these 23 chromosomes, 23 pair of chromosomes, 22 are autosomes and one pair is allosome, that is X and Y. Great. Okay, so now this zygote is formed. Still remember, this everything was happening inside, right? So this zygote is formed. This will be one cell, and outside there will be second polar body, which will degenerate eventually. And next to it is this zona pellucida, this fertilization membrane, and corona radiata. This everything is still there. Slowly they will degenerate. Okay, and this zygote will develop into embryo. Fine. And during this whole process, remember, this whole process has occurred into the ampulla, okay, or to be precisely at the junction of ampulla and isthmus into the fallopian tube. Now what happens? The zygote will gradually starts to develop into the embryo. Meanwhile, it will continue its journey. Through the fallopian tube towards the uterus. Okay, so remaining part of this we are going to see in the next lecture, where we will be talking about the embryology, embryogenesis. We will study how this zygote will divide, okay, to form embryo. We will look at different stages of embryo, and then how this embryo develops into fetus and organogenesis and the rest of the things. Okay, so stay tuned and stay safe. With that, thank you very much to be with me in this lecture.